Hi everyone, and thank you for joining. The work that I'm going to present you today is a collaboration that started in ULB in 2016, and most of the results that I will show you have been published last year in this Journal of Biogeography. But we have two ongoing follow-up analyses of this study, and I will present you some preliminary data from that. And you all know that tropical rainforests are the most diverse and richest species of ecosystems in terrestrial ecosystems. And the diversity that we see now in these ecosystems has been the effect of many factors shaping throughout its evolutionary history. And during the last decade or so, many analyses that have been incorporating this meta-analysis that incorporate ecological data with full phylogenies have now been starting to understand how these different factors have been shaping this diversity. So for instance, now the current distribution of the tropical rainforest is in these three main blocks, so the Neotropical, Africa, and the Indo-Pacific. And unlike the Neotropical and Indo-Pacific, there is a sharp decrease in tree species. So the numbers you see here is the estimates for tree species in each of these regions. And on the top graphs, on the y-axis, you have the estimated area since around 50 million years ago, and how that area has been reduced or changing across time towards the present. And it's quite constant the change when you compare the two other areas with the area reduction in Africa. And so the work that I'm going to present you today has two components. One is the forest per se, so the ecosystem, the biome per se, that is the shrinking forest. And the other is the inhabitants, that will be the tree species that are living in those ecosystems. So if we go and start with the shrinking forest and going back to the history, the long time history, you can see how, from a geological perspective, how this area, the Guineo-Congolian region, came to be so reduced to the present. Since mainly from the Middle Eocene, there was a sharp reduction in the area that was covering this ecosystem. But it's not only the overall trend in reduction of diversity, especially during the last glacial maximum, there has been these cycles of contraction and expansion of the forest. So in addition to this overall trend of reduction of the area, you have some areas where you have expansion and then contractions. So those species that were adapted to these ecosystems have these options to following the movement of the forest per se, or being faced to new habitats and then exploit those habitats if they were able to do it. So the other component that I was mentioning to you, the tree inhabitants, it's in the picture that I showed you before of the number of the species was overall estimates for tree species, considering all families. But if you look at different families, one of these groups, the Tarioidae, that is one of the 6 subfamilies that are now in legumes, is an odd group within this trend. It has higher species diversity in Africa compared to the Neotropics or the Indo-Pacific. So this group is a good system to start studying how these effects have affected the diversification of this group. But if you are into tropical fruits and you know Tamarindus indica, this is one of the species that belongs to this group. But in order to really measure what is happening in this effect between the changes in the area and diversification rates, it's better to use a group that is very specific and endemic to Africa. And so for this study, we selected one lineage within this subfamily, that is the Berlinia clade, that has 16 genera and about 190 species. And this group is renowned because if you see this Serengeti or African biomes, the savanna biomes, these trees that you see here, some of them, or the main component of this biome is a species from 3 genera that belong to this group. And some of the hypotheses of how the evolution of hominins are based on these changes in biomes, and species from this group were involved in the forming of these biomes. So the main question of this study was to assess what has been the effect of this reduction of area, both in the long term, this steady decline, as well as this short term, more recent expansion and contractions of the area of the forest. So basically, we had 2 hypotheses. One is that as you see a decrease of the area suitable for this species, there was an increase in extinction rates. So the overall clade reduce their diversity and they become extinct or not diversify. And the other hypothesis is that as this contraction reduced, there was a creation of new habitats and also fragmentation of the habitat per se. So allowing both allopatric and ecological speciation opportunities. So if the lineages were able to shift to habitats or to new interactions, they will be able to diversify and increase in diversity. So the first thing that you need in this case is a solid and framework to do it, and this is a phylogenetic tree. And so the first step that we did was to generate this tree for the group, and we use an exome capture of 289 genes. And now we know a good estimation of the relationship of the group, and we have identified these 3 main clades in the Berlinia clade. And having this framework, you can also put the age of fossils in the analysis, and then you can estimate this branching of the tree associated with an age. And by doing that, we know that the clade per se originated more or less in around 15 million years ago, so in the Miocene. And what we see, the diversity that we see now at the genus level in this group mainly originated during that time. So it's very ancient lineages that are present now in the area. And this is quite consistent with what people has been finding in other groups, like in Araceae or palms, that most of the genera diversify in Africa in the Miocene. But something that is quite unique and particular to this group is that we found some genera that diversify more recently in the Pliocene and Pleistocene. And because for some of these genera, we have already full sampling at the genus level, so all the species included, we know that there has been also diversification in these genera and in the Pliocene. And this is something that is not commonly found in Africa. So the next analysis that we perform is are these clades increasing, decreasing, or maintaining a steady diversification rate across the time? So in this, you use these birth date models and a maximum likelihood framework to assess what has been the trend in terms of speciation. So considering only the evolution and the accumulation of species through time, we found that there has been an increase in diversification rates in the group. And then once you have that, you can associate that with any environmental factor that you want to test. In this case, we wanted to test this forest area reduction. So we use this geothermal estimation of area covered since the past to test if there was any association between the reduction of the forest and the diversification rate at the clade level, so the entire Berlinia clade. And we found that the best fitted model is an environmental dependency with exponential increase. So there is no association as the forest was decreasing. There was not a reduction of diversification in the group. So the reduction of the area available for this species promoted diversification. And to understand more how this lineage was still maintaining this high speciation rate, despite that the forest was decreasing. So once you have this phylogenetic framework, you can estimate or figure out what was the ancestral state, ancestral habitat of the group. And we found that it is the forest, and that this, when we analyze at the genus level, so each genus, we analyze if all the species were still living in the forest, or there was some species that have changed habitat types, and codify this as a labile trait in the phylogeny. And we found that this capacity is not homogeneous distributed in the tree. It's asymmetrically distributed, especially in lineage C. And we found that these genera that now are the main structure of the Miombo savanna evolved 3 times in these 3 different genera. And something additionally that we found is that this is the same phylogenetic tree, but now each point in the tree represent 1 species in the analysis. So what I want to highlight is that it's not only the capacity to shift that is asymmetrically distributed in the clade, also the mechanisms by which each lineage shifts habitat is a bit different. Like in this genus Brachystegia, the ancestral state within the genus is forest. So the lineage, or when the genus evolved or originated, the ancestral was adapted forest. But then there was 1 shift to exploit this savanna. Yeah, and after that, it simply diversified in many species. And in other genera, you have multiple individual single shifts, like in the Berlinia, where we found 3 independent shifts to the savanna. So there are different mechanisms that were employed by this group. And finally, because we now have one of the genus that we start sampling more completely was this Antonota with 17 species. And when we were analyzing, Sandra was checking the distribution of these species, we found that all those species that were widely distributed in the Guineo-Congolian region were nested in 1 single clade. And when we analyzed 1 species, Macrophylla, across the landscape, we found these clusters of diversity, and FSTs indicates that these 2 groups in the Lower Guinea are more recent and probably exchanging, and that fits with our phylogenetic analysis, where these areas are located in a phylogenetic framework. So we have identified these areas that are barriers to gene flow in this group. So to conclude, what we see now, or what we have in this tropical rainforest in Africa, is an all genus level diversity. Most of them, 75% before the Pleistocene. And the ancestral group, the ancestral habitat in the group is the forest, and it have a asymmetric distribution of this capacity to shift habitat types. And something that is not common in Africa is that we found recent diversification in some genera. So overall, the reduction of the forest area has promoted diversification, probably by different mechanisms. So some genera still diversify, but without shifting to habitats, probably exploring different pollinators or dividing the niche in a different way, while others simply exploited the new habitat available, in this case, savanna, more dry environments. And with that, I would like to thank you for your assistance, and people that help us during the development of the debate and provided samples and funding from NIBU. And we'll be happy to take any questions.